All right, welcome to part 2 of Chapter 7. Here we want to start out by reviewing aerobic cellular respiration and then looking at some inhibitors, things that can inhibit the process. Then we want to look at how, we talked about in cellular respiration, how we metabolized simple sugars that we can convert to glucose, but we want to look at how we would metabolize proteins, lipids, and complex carbohydrates too. Then we're going to look and see how catabolism and anabolism are tied to each other. And then we'll look at aerobic cellular respiration, and then get into fermentations and specifically look at lactic acid fermentation and alcohol fermentation, and then finally finish up by how we regulate cellular respiration. Well, first of all, recall that in aerobic cellular respiration there are 4 parts. Glycolysis gives a little bit of ATP, but really is just preparing things to send them into the mitochondria. We're going to spend that 2 ATP. And then in pyruvate oxidation, we're getting some more electrons and protons and converting the pyruvate to a 2-carbon group that can go into the citric acid cycle. It needs to be escorted by coenzyme A. The citric acid cycle, or Krebs cycle, is only going to give us 2 ATP total, but we're going to harvest a whole bunch of protons and electrons. And when we put all those protons and electrons through the electron transport chain, couple it with a process of chemiosmosis, that's where we're going to get the most AT, about 34. And so in a eukaryotic cell, we figure about 36 ATP when we use the mitochondria and fully oxidize glucose. But there are things that can interrupt these processes, especially the electron transport chain and chemiosmosis. Rotenone is a chemical that shuts down the shuttling or redox reactions of passing the electron in the electron transport chain in fish. And it's used a couple ways. Let's say that there's a lake or a pond that has fish in it that we don't really want there, carp and suckers and maybe the grass carp. That's a big problem. Well, we can poison the water with rotenone. It'll kill all the fish that are there. And then we can scoop them up with nets and get them out of the water, and then we can restock the lake or pond with fish we want to use. And that's how rotenone is commonly used. Cyanide, you've probably heard of that poison. Every spy had their little cyanide tablet in their ring. But what cyanide does is it shuts down the end of the blocks electron transport, the end of the electron transport chain, so that we can't utilize the oxygen. So we're not getting the full energy or ability to we're not getting the full energy or ability to make ATP. Carbon monoxide does the same thing. This is why it's so important not to let carbon monoxide build up in closed spaces, because it shuts down your electron transport chain. Now your cells can't generate enough ATP, and then you kick it. So you want to be careful with that. DNP stands for dinitrophenol. What dinitrophenol does is it increases the permeability of the inner cell or inner mitochondrial membrane, and so instead of H plus or protons accumulating, many of them leak back through the membrane. And so instead of going through ATP synthase, where we can make ATP, they're just leaking back into the matrix. And so this is going to dramatically reduce the amount of ATP that's produced. They used to think that DNP would be a good weight loss drug because it would keep you from getting as much energy as you could get from a molecule of glucose. And so you'd lose weight. But the problem is there's some side effects, neurological side effects that go with that process, so you don't want to do that. Oligomycin is an antibiotic that targets ATP synthase in bacteria. Even though your cells and bacterial cells use the same process, the enzymes are slightly different. And so an enzyme like oligomycin that targets ATP synthase in a bacteria is not going to target ATP synthase in you, because your enzyme is just a little bit different than the bacterial enzyme. They do the same thing, but their structure is a little bit different. So, discussion question. Cyanide blocks final transfer of electrons to oxygen. What will happen? And so we'll kind of kick on this for a little bit, give you a second to think. When you're ready, kick it back on. Okay, well, we're probably going to see a reduced ATP yield because we're not going to be able to push as many protons that can go up. Only glycolysis Krebs can happen. So we're going to get our 2 ATP from glycolysis. We spend them to get in the mitochondria, so they're gone. And the Krebs can happen, but we're not going to be able to harvest the electron energy there. So that might be miswritten. Only glycolysis can happen. No, we could get well into the Krebs cycle and partway through electron transport, and not 0 ATP because some of the protons are getting pumped, just not the whole. So I would say A is the best answer for this, slightly reduced ATP yield. Now, at this point, we've looked at using a monosaccharide, glucose, for generating ATP. What are we going to do with other nutrients? Well, if it's a disaccharide or a starch, like edible plant starch or glycogen, we're going to break those down into monomers of glucose or other monosaccharides that can be converted to glucose, and then they're going to go right into this pathway. Okay, so there's just going to be other reactions going on here to convert these into glucose. If it's a fat, it's probably a triglyceride that we're using for energy production. And triglycerides are a 3-carbon glycerol, and then with 3 long-chain fatty acids. Well, the 3-carbon glycerol is going to be inserted into, enzymes are going to insert that into the middle of glycolysis when we first had 3 carbon molecules. The long-chain fatty acids are going to be chopped up 2 carbons at a time by a process called beta oxidation, and we're going to make a bunch of acetyl-CoAs. So if we've got a 10-carbon fatty acid, we can make 5 acetyl groups. We're going to turn that into 5 acetyl-CoAs and go around the Krebs cycle 5 times. So you can see fats, you've got 3 long-chain fatty acids and a glycerol. You can have a lot of energy stored in these molecules in terms of releasing electrons and protons to generate ATP. If we have a protein, most of the time proteins are not going to be used to make ATP because we need amino acids to build our own proteins. So we're going to take proteins in our diet, digest them into individual amino acids, and then most of the time those amino acids are going to be recycled to make other proteins. But what if you have a diet that's all protein and you need to break those proteins down to make ATP? Well, what we're going to do is convert the proteins to amino acids. We're going to get rid of the amino group, either completely get rid of it and turn it into urea that's lost in your urine, or we're going to store it on a molecule somewhere else in the cell and save it so that we can use it to make molecules that need a nitrogen. And then we're going to take what's left of the protein, its carbon skeleton, and we're going to chop it into 2, 3, 4, 5, or 6 carbon pieces that are going to fit into glycolysis, the oxidation of pyruvate, or the Krebs cycle, wherever we end up using it. So, that's what we would do with proteins. Now, here's the thing, is all of these reactions go in both directions. At this point, we've looked at breaking down the large macromolecules to generate ATP, but we can also use the intermediates as raw materials to build these molecules. And so what you have to realize is because the needs of the cell are constantly changing, sometimes we're putting things together to build the big molecules. Sometimes we're breaking them down to generate ATP. It just depends on what the cell's needs are, and those are constantly changing because the environment is changing. So the big point on this slide is that all of these metabolic pathways go both ways. The macromolecules can be broken down and used in the pathway to make ATP, but we can also take ATP and take intermediates from these pathways and put them together to make the macromolecules that the cell needs. All right, so we used in aerobic cellular respiration, we used oxygen as our terminal electron acceptor, and the reason is because oxygen is the best. When oxygen is used as a terminal electron acceptor, we get the most ATP from our electrons and protons in oxidative phosphorylation. That's why all multicellular organisms are aerobic. They may have times when parts of them are anaerobic, but for the most part, aerobic respiration is the key to being a large multicellular organism. But there are some bacteria, single-celled organisms, that can use different terminal electron acceptors other than oxygen. They can use nitrate and convert it to nitrogen. They can use manganese and convert it from manganese oxide to manganese. They can use iron 3 and convert it to iron 2, different charge. They can take sulfate, convert it to dihydrogen sulfide. They can take CO2 and convert it to methane. And the order this is going to occur is shown. First, we're going to use oxygen, and then the nitrate reducers are going to kick in, and then the manganate reducers, then iron reducers, and the sulfate reducers, and finally the methanogens. These are all bacteria. They're prokaryotic organisms, and they have evolved to utilize these pathways because other things are using oxygen. And so, if you can use carbon dioxide as your terminal electron acceptor, you can survive where none of these other things can. And that's the key to how organisms were able to evolve, is if there's a place with nutrients that everything else can't survive, but you can, then you get all those nutrients. And those nutrients go for your growth and development. Okay. And one place where this is really used is in bioremediation of toxic materials. Many times, the pollutants that have been released are going to shut down aerobic respiration, or they're too concentrated to be tolerated by aerobes. And that's where the anaerobes can be used to help break down. And when you remediate something, you bring it back to the way it's supposed to be. And so, fixing human mistakes and contaminated areas or spills, more and more we're looking for bacteria to do this for us, because we need to clean up the environment so that it isn't toxic for us to survive there. Fermentation is another anaerobic process, but it doesn't use the electron transport chain and oxidative phosphorylation. That's the difference. Basically, we're only getting energy from glycolysis. So you go through glycolysis because you don't need oxygen for that. Glycolysis occurs where oxygen is present, but if oxygen is present, you're going to keep going onto the other parts of aerobic respiration. When the oxygen is all gone, that's when you only ferment. And you get your 2-ATP from glycolysis, but you don't get much else. And the problem with just getting energy from fermentation is that you can't let pyruvic acid or pyruvate accumulate because it's toxic. So what ends up happening is we go through glycolysis and we end up with our product from pyruvate. But then what we're going to do is we're going to take our NADH plus H plus and we're going to donate those electrons and protons back to create something else that's less toxic than the pyruvate. And now we can keep going through glycolysis, getting our 2-ATP at a time. And the two main types of fermentation we want to look at are lactic acid fermentation and alcohol fermentation. Okay. Again, fermenters are only getting ATP from glycolysis, 2 ATP at a time, but they can't let pyruvate accumulate. So they convert the pyruvate back into a fermentation product, and that way they can keep going through glycolysis. Now, this is way less efficient than respiration. Okay, way less efficient, but it allows them to keep going. Interestingly, cancer cells are locked into glycolysis. They don't, when a cancer cell is actively growing and metabolizing, it's not going through pyruvate oxidation and the Krebs cycle and oxidative phosphorylation. And the reason is because it needs the intermediates from glycolysis to build all the proteins and macromolecules that the cancer is going to need to grow as fast as cancer cells grow. We're going to see later when we talk about mitosis in the cell cycle that cancer cells are cells that are just growing out of control. And when you're growing and just continually doubling your numbers all the time, you don't need ATP as much as you need the intermediates to build the large structures you need to replicate cells. In lactic acid fermentation, we're going to take our pyruvate and we're going to put the electrons and protons back to convert pyruvate to lactate. Okay, so here's pyruvate, and it's got a carboxyl group and a carbonyl group, and now we've turned that one carbonyl group into a hydroxyl group. Lactate or lactic acid is less toxic than pyruvate. This occurs in your muscle cells. When you're active, and you're breathing, you're bringing oxygen in your bloodstream, which goes to your cells. There's always some oxygen stored in the cytoplasm, and there's oxygen stored in other places in the cell. But if you're highly active, you're going to use that up pretty quick. And then what happens is, you hear the old thing, the runners hits the wall. Well, that's where you make the transfer from aerobic respiration to fermentation. And so you keep going through glycolysis to get your 2 ATP at a time, so your cells can keep going, but not at their peak level. But you can't let pyruvic acid accumulate, so you convert it into lactic acid. And that lactic acid accumulates in your muscles, and that's what makes you sore. And if you're a highly conditioned athlete, as soon as you're done whatever you're doing, you repay the oxygen debt in your liver cells, you turn the lactate back into pyruvate, and you burn it up to get energy for the cells. And you don't feel the pain for very long. If you're not a well-conditioned athlete, it may be weeks before you slowly leach all that lactic acid out of your muscles through your liver and break it down back into pyruvate. We also do this process. There are bacteria, especially in food production, where we're using this fermentation process to create foods that have flavors or textures that we like. We use lactobacilli that are capable of lactic acid fermentation to turn milk into yogurt and sour cream, to turn cabbage into coleslaw or kimchi, to turn cucumbers into some types of pickles, and to turn milk products into some types of cheeses. The enzyme that does this is an enzyme called lactate dehydrogenase. I don't need you to know that for the exam, but it's not a bad thing to realize that all these enzymes control a specific reaction. This one controls putting the electrons and protons back onto pyruvate to make lactate. So this is big business. In food production, this is happening in huge factories where they're producing, probably worldwide billions of dollars worth of economy making these foodstuffs. Alcohol fermentation is another important chemical reaction. There are some yeast species that are capable of doing that. And what they do is, again, they when you first add the yeast to whatever they're going to break down, there's a little bit of oxygen there. And so they're capable of using the oxygen, going through oxidative phosphorylation, getting a lot of ATP. But as soon as the oxygen is used up, they can only undergo glycolysis. And we did this in the alcohol fermentation lab, where we took our sucrose and our yeast, and we let the yeast break down the sucrose, and they right away used all the oxygen that was there, and once it was gone, they started releasing CO2. That was the gas that we measured. And here in beer fermentation, you can see the massive amounts of CO2 that are being released up on the top of the malt. But then what we're going to do is we're going to add those 2 electrons back, because the intermediate of this is acetaldehyde. Acetaldehyde is toxic, and so we don't want it to accumulate. So after we decarboxylate, we kick off a CO2, we're going to add back the protons and electrons to convert acetaldehyde into ethanol. Now, eventually you get to a percentage of ethanol, about 18%, that's toxic to the yeast cells. That's why beers and wines are kind of limited in their ethanol concentration. If you get too much ethanol, it kills the things that are making the ethanol. Okay, but this is big business. We produce billions and billions of dollars worldwide are generated by the fermentation of ethanol. Part of it for consumption, but the vast majority of ethanol is not used for consumption. The gasoline in regular gasoline is 10% ethanol. In the flex fuel E85, it's 85% ethanol. In many factories, they'll take ethanol as a raw material and let microbes or otherwise modify it to make other things. For example, there are bacteria that will oxidize ethanol to form acetic acid. 1.5% acetic acid is vinegar. And think of all the things that vinegar is in. Well, they start with ethanol as a raw material. So this is a huge reaction. And frankly, this is probably one of the oldest evidences of biotechnology in human society. Almost every population that's been studied has found a way to ferment grains or fruits to produce alcoholic beverages. Okay, so this is as old as civilization is, human civilization. So if I look at the overview of cellular respiration options, I'm going to go through glycolysis, and then if oxygen is present, I'm going to undergo the Krebs cycle and either use oxygen as terminal electron acceptor and get my maximum ATP from aerobic respiration, or use some other electron acceptor other than oxygen, still get more ATP than I get from glycolysis. If I don't have any oxygen present and I'm a fermenter and not using something else as an electron acceptor, then I'm just going to go through fermentation and get my 2 molecules at a time from glycolysis. But realize that many of these fermentation end products we have a use for, and so we encourage the fermentation to occur to generate the end product. Okay. Now, all cellular processes are regulated. We don't want a cell making ATP that we don't need. And when that happens, that's when you're going to have unexplained weight loss, which is that symptom, and that means your cell's burning energy that it doesn't need to burn. I mean, if you drive to class and you're sitting in a classroom in a building or in the lab, you didn't leave your car running in the parking lot because you're wasting energy. And living things don't want to waste energy either. When that happens, it's an indication something's wrong. So we want to have different mechanisms that control cellular respiration. And there's a variety of them. One of the big ones is hormonal control. In our bodies, we have an endocrine system. And those of you going on to A&P later, you're going to learn a lot about that. But endocrine system produces hormones, one of which is insulin. And insulin determines whether or not our cells can pull glucose out of the bloodstream. You have an insulin receptor. The insulin binds to it. That causes a vesicle to release a protein called GLUT4 or glucose transport protein 4, which then allows glucose into the cell. People who don't produce enough insulin or any insulin that are diabetics or have an insulin receptor that's not working very well can't send this to the cell membrane. They struggle to go through glycolysis. Their glucose ends up being lost in their urine, which causes problems with the kidney. And, so they have to supplement they have to take insulin injections or watch their diet to not have too much glucose. There are also enzymes that can be reversed or irreversed depending on where the equilibrium is, so that we can cause ATP to be formed or use the intermediates to build the macromolecules that the cell needs. So there's constant oversight on these. And of course, a lot of these are based on feedback mechanisms, and we looked at feedback inhibition already. If we've got a lot of end product, there's no sense in making any more of it. When we run out of end product, that's when the pathways resume. And then pH changes. pH changes can influence the enzymes and what they do. So if we're undergoing lactic acid fermentation, that lactic acid builds up, it lowers the pH, and now the enzyme may not function. So there's a variety of ways that cells can regulate whether or not they're going to undergo any of the forms of cellular respiration to produce ATP. And if we're going to, want to make a connection to this, think about your body. The average person needs about 2,200 kilocalories per day of energy, but that's going to vary from person to person. The bigger you are, the more kilocalories you're going to need. About 75% of those are just your basal metabolism used to maintain a healthy body. So, and you're constantly using ATP when you're asleep, when you're awake, when you're doing things, when you're at rest. The other 25% is used to power physical activities. So when you become active, and of course some of these, you're running 8 to 9 miles per hour. That's a pretty good jog. 10 miles per hour would be 6-minute miles. So 9 miles per hour is probably, a 7-minute mile or something like that. That's pretty quick for a distance. You're using a lot of kilocalories per hour. Okay, dancing fast compared to dancing slow, right? Driving a car uses more than sitting at a desk writing, but not near as much just walking, right? And you pick up 1 mile per hour, and you gain a little more. If you can swim slowly and burn even more. So and there are a lot of online calculators and indices that can help you to determine how much energy you're burning in an hour. Realize that a pound of fat has about 3,500 kilocalories of stored energy. So if you want to lose a pound of fat, you've got to shave 3,500 calories out of either what you take in or what you burn off. And so if you could lose, an extra 500 calories a day of the difference, in 7 days you should lose a pound of fat. And that's the end of this section, or this particular lecture. Again, here are the key terms you should be familiar with for the exam. And here are the key concepts that you'll want to be ready to field questions about when you take the test.